Hi everyone, welcome to the penultimate neurobiology session of the Varsity Sci Symposium. So it's the last day of the symposium today. We hope to see you for our next session. But first of all, for this first session today, we're having a neurobiology theme. So the speaker is Christopher Mak from a medical student at Cambridge. He will be speaking about evidence supporting and refuting the effects of probiotics in treating Alzheimer's disease. I will be moderating the Q&A session after his talk. So if you're on Zoom, feel free to ask your questions in person, send them to the chat or send them to me privately. Or if you're on YouTube, feel free to leave your questions and comments throughout the talk. Without further ado, I'll now pass the floor to Christopher. Thank you.
Hi, I'm Christopher Mak. I'm a medical student at Cambridge, and today I'll be sharing my talk on how probiotic supplementation for age-related neurodegeneration might ameliorate its pathology. And this is done in collaboration with the CUHK, the Chinese University of Hong Kong, and also Cambridge University Genetics Society. Now, some background on what Alzheimer's disease entails. Alzheimer's disease is a disease of memory, where memory is essentially a faculty of the brain in which data or information is encoded, stored, and retrieved. And there are many models to which memory is described. First off, initially by Atkin and Schifrin through the multi-store model, then by Badley and Hitch with the working memory model, and finally with Squire and the long-term memory model. And essentially, what these models show that in long-term memory, the hippocampus is central to remembering and even amnesia and pathology. Age and memory are intimately linked. When you think of memory and how it relates to age, you think of memory loss. And what memory loss means is retrograde amnesia, forgetting stuff in the past. And apart from this, other problems arise with age, such as a decrease in fluid intelligence and also a decrease in cognitive abilities. And all of these ultimately are intimately tied to the hippocampal atrophy, the neurodegeneration in the hippocampus, where there's a correlated increase in the lateral ventricle volume of the temporal horn, and also a decrease in gray matter. And obviously, while these diseases will have a clinical onset around the time of elderly, around 50 to 70 years old, oftentimes. However, the pathologies often start in middle age, around 40 years old, but often in preclinical stages or even at a less phenotypic stage, such as at mild cognitive impairment, MCI, before ultimately progressing to dementia. There are various hypotheses for Alzheimer's disease, and the first and the most famous one is the amyloid hypothesis. And this essentially is a process where the APP gene encodes the amyloid precursor protein. However, instead of being cleaved by the alpha secretase, it is instead cleaved by the gamma or beta secretase, producing A beta 40 or A beta 42 peptides, which are prone to aggregate as oligomers and ultimately form amyloid plaques in the extracellular space. And this extracellular aggregation results in insoluble precipitates, and with them being insoluble in the brain extracellular phase would lead to neuroinflammation, oftentimes affecting microglial function, and ultimately are neurotoxic since they can't be cleared and result in a pro-inflammatory state neuroinflammation in the brain. However, this hypothesis by itself does not describe the entire story, and there are certainly limitations in how this hypothesis is used. For example, in a paper in Nature, it described how drugs targeting this hypothesis often fail, often don't work, because clearing the A beta plaques don't simply ameliorate Alzheimer's. Rather, there are other hypotheses, other mechanisms involved. And the second most common mechanism is the tau hypothesis, where tau forms neurofibrillary tangles, which are essentially a pathology stemming from intracellular aggregations of tau. And these taus are pathogenic because they are hyperphosphorylated and ultimately form tangles which induce neuroinflammation and ultimately lead to neurotoxicity. And look at here, together with amyloid plaques and NFTs, they will ultimately result in a decrease in cortical gray matter, particularly at the hippocampus, and thus result in memory loss. Other common hypotheses include the cholinergic hypothesis, where there's a loss in cholinergic neurons in the basal forebrain. This part of the forebrain near the prefrontal cortex, particularly at the nucleus basalis of Meynert, and loss of neurons there, also due to the loss of cortical gray matter, will lead to an impairment in memory formation and learning. And obviously, the newest hypothesis is the microglial hypothesis, which essentially microglia are cells which are macrophage-like. They are found in the brain and CSF. For simplicity, there's the M1 and M2 phenotype, but obviously there's a lot more. And it is the inability of microglia to clear amyloid plaques, and also their chronic activation in the brain, which lead to neuroinflammation and ultimately lead to memory loss and Alzheimer's disease. And the propensity of these microglia for chronic inflammation and for them to not be unable to clear amyloid plaques is dependent on the TREM2 variant, which was studied by Professor St. George Hislop in the University of Cambridge. And obviously, there are other hypotheses: calcium MDA, neurovascular inflammation, metal ions, lymphatics even. But the point of these many hypotheses is to illustrate that there's always a certain biomarker that is adversely affected in the brain. And what we're trying to study here through the study is to see how, by supplementation of the probiotic, is it going to mediate the gut microbiome and hence the gut metabolic profile, and how the metabolome, the metabolites from the gut will through the gut-brain axis, through the blood-brain barrier, affect brain function and ultimately affect memory formation in Alzheimer's disease. Now, what is the gut microbiome? The gut microbiome is a new area of interest starting from around 2013, 2014, whereby it is characterized by the bacteria colonies and microorganisms residing in the gut, the GI tract, particularly in the colon. The gut microbiome profile of a person varies with age, health, and of course diet, and an aberration in the gut microbiome, which is also known as gut dysbiosis. This meaning aberration, and biosis means the gut microbiome. Gut dysbiosis is often observed and correlated with physiological diseases, and is currently a target for different people, and it can be changed through the use of fecal microbiota transplants, through the use of probiotics, which is what we are studying today, and even prebiotics and symbiotics, and it is adversely affected by antibiotics. Obviously, the pathways involved with the gut-brain axis and how the gut microbiome ultimately leads to neurodegenerative pathologies is very complex and multi-staged. It involves neuronal factors, endocrine factors, cytokines from immunological factors, but ultimately they all converge on a similar mechanism, whereby it is modulation of the gut microbiome, such as the amelioration of the gut dysbiosis, results in different metabolic pathways, the metabolome, and how the change in metabolome ultimately leads to would pass through and cross the blood-brain barrier, and this would ultimately affect the physiological state in the brain, particularly the hippocampus, and result in Alzheimer's disease, which modulates dementia, memory loss, and etc. In typical aging, particularly in elderly, gut dysbiosis is a common occurrence, and it is due to an imbalance of commensal bacteria species in the microbiome in the gut, and this leads to a change in metabolome, of course, and the change in metabolites in the gut would disrupt the gut mucosal membrane. This is described by the leaky gut hypothesis, and ultimately these metabolites would reach the brain by crossing the blood-brain barrier. And particularly in elderly with often an aberrant immune system, which is often weakened, and this results in an increase in amyloid peptides. Bacteria producing lipopolysaccharides would induce neuroinflammation, and oftentimes this is correlated and associated with a decreased synthesis and secretion of neurotropic factors. And these all result in a phenotype of Alzheimer's disease. Obviously, these are all really complex, but there's always a common theme. And the common theme for today's study on how probiotics can affect neurodegeneration pathology is via the gut, immune, and brain axis. There's currently evidence for how probiotic administration can ameliorate Alzheimer's pathology. And in preclinical in vitro studies, is shown in vitro and in animal in vivo studies, is shown that probiotic supplementation can improve animal cognitive performance and also behavioral performance. And the most common bacteria strains of choice include Lactobacillus and the Bifidobacterium genera. And a review by Westphal, which is quite famous within the field, showed only animal studies indicated only in animal studies there's evidence indicating that probiotic supplementation has ameliorated neurodegeneration. Therefore, the object of this study today is to elucidate whether probiotics can efficaciously ameliorate age-related neurodegeneration. Now, methods. To best study this, we have done a systematic review, searching through 6,000 studies across 5 databases: Medline, Embase, Scopus, Web of Science, and Cochrane Library, where we have searched through the bibliographies of the reviews involved. And we have selected in vivo studies only, including human and animal data. And we have looked at Alzheimer's disease, Parkinson's disease, frontotemporal dementia, and even mild cognitive impairments. And we have excluded studies which has not specified probiotics and other unrelated diseases. We have analyzed the data in both a qualitative way and a quantitative way, and quantitative analysis is done through these measures. For human studies, humans are analyzed through the MMSE, the Mini Mental State Examination, which is a questionnaire assessing their cognitive performance. And animal studies are assessed by these 3 measures: the Morris-Water maze escape latency, Y maze. Y maze is a maze where they have 2 choices, and the percentage of alteration between the 2 choices in a maze is the measure of behavioral ability. And finally, passive avoidance, which is essentially a measure of latency on how they passively avoid a certain choice. And this meta-analysis or systematic review is done through RStudio and RevMan. Risk of bias is assessed through the Cochrane Risk of Bias 2.0 tool for human studies and the SYRCLE tool for animal studies. Now, let's go to the results of our study. Through collating these amount of studies, we have found a total of 5 human and 20 animal studies, of which 25 of these can be used with qualitative data. First, analyzing the qualitative results, we have found that in terms of gut microbiota alterations, in cases where there's the probiotic supplementation, there's a reversion of gut microbiome dysbiosis back to a healthy gut microbiome, and this is associated with an increase in acetic and propionic and butyric acids. It's also found that there's a positive neurotrophic effect where there's an upregulation of brain-derived neurotrophic factor DCX, phosphorylated Krebs protein, ghrelin, leptin, GIP, and GLP incretins. There's also an increase in anti-inflammatory effects where there's an increase in IL-10 MCPs and a downregulation of TNF-alpha. There's also antioxidative effects where there's an associated decrease in MDA, SOD, GPX, and catalase. All of these are enzymes related to oxidation. And of course, reduced microglial autophagy and apoptosis, where there's an upregulation of OGG1, decrease of cathepsin B, and increase in cathepsin L, all associated with microglial activity. And finally, there's a decrease in amyloid beta plaques, which in the hippocampus, shown through immunohistochemistry findings in the mice and rat brains. Collating all the quantitative data in humans, collating the MMSE scores in humans, and we have assessed them through comparing placebo and probiotic populations, I found that there's in general a positive improvement in MMSE score. And what this graph shows is that in the cases of Alzheimer's disease, where they all have significant and severe mental degeneration and retardation, the supplementation of probiotics have significantly improved their mental faculties and cognitive abilities. However, the same change has not been seen in the cases of patients with mild cognitive impairment. We've done the same in animals, assessing the animals by Morris-Waterma's escape latency, where the quicker they escape, the mice have better cognitive ability, and if the mice escape slower, the mice has more memory problems. And comparing control against treatment across 30-something studies, we found that there's in general 14-second benefit with probiotic supplementation across Bifidobacterium, Lactobacillus, and other species. And this shows that by supplementing probiotics to mice and rats with Alzheimer's disease, we can essentially improve the cognitive performance and at least ameliorate the Alzheimer's pathology. A similar result is also seen in animal studies collated through Y maze, where the Y maze changes in spontaneous alterations also show a positive improvement. And the same finding is also found in studies collated through passive avoidance. And there's also around 108-second improvement in passive avoidant latency, suggesting that probiotic supplementation, probiotic treatment can improve the neurocognitive functions of Alzheimer rats. Now, breaking down the previous graphs into various subgroups and let's see what kind of confounding factors might be the case. By breaking down them as per dosage of probiotic supplementation, we have found that in higher dosage, at least in human studies, there's a better effect in amelioration. However, the same effect is also found in Iranian populations, where studies done in Iranian population has shown to be a better effect. However, the same copy of the same exact effect is also found in mean age and disease. And this is a bit suspect because the change are exactly the same. And this, I believe, is down to the fact that the research into gut probiotics and Alzheimer's disease is only done by two separate groups, one in Japan and one in Iran. And this had led to a bit biased results, and I'll get back to the bias later. For animal data, however, the results are rather uniform, and all of them had a significant difference of 0.05. Now, taking the previous data and meta-regressing them through RStudio, we have found that if there's a significant effect and difference when with age and MMSE score by probiotics. Essentially, this graph shows that with increasing age, the effects of probiotics in improving Alzheimer's disease is stronger. And what this graph shows is that perhaps the dosage, perhaps the application of probiotics occurs in an age-dependent manner. For animals, however, there's no covariates, which shows there's a significant difference, essentially suggesting that there are no confounding variables and that the other effects such as dosage, or even the supplementation frequency has no significant difference, essentially showing that as long as you supply probiotics, regardless of what you do, duration, dosage, etc., there'll be an effect. And as you can see, all the confounding trends have a mild slope, and there's always a more than 0.05, essentially insignificant difference with all these trends. Likewise for Y maze, and also the same for passive avoidance. There's no significant difference, and there's no significant correlation. Assessing the bias of these studies, we have looked at how these studies differ, would scatter across a funnel plot, and human studies are found to be scattered outside the 95% confidence intervals. This is Beck's funnel plot, and it shows whether the studies are biased, whether they're publication bias in the studies or not. And clearly, with a bidirectional distribution exceeding the 95% confidence interval, there's a certain amount of bias, and this is also down to the fact that the studies are only produced by 2 groups, one in Iran and one in Japan. For animal studies, however, the results are more believable and more robust, since most of the studies lie close to or within the 95% confidence interval, and that the spread of studies is quite uniform. The same is also found in wire maze, and also a similar spread is also found in passive avoidance. We further assessed the risk of bias through assessing the methodologies of the various studies through use of the Cochrane's ROB2 in humans. And while most studies have a low risk of bias, there's a few studies which contribute to a high risk of bias, and I believe it's these studies which leads to a skewed results in human data. And the skewed results is due to some errors in methodology. There's a lack of allocation concealment in 1 or 2 studies, a lack of blinding of participants in 4 or 5 studies, and also ultimately an unclear selection of purported results, i.e. The results and methods are often not reported. Looking at animal studies using the SYRCLE tool, we found that most of the studies have an unclear risk of bias. We cannot assess whether it's high or low. And this is mainly due to a few issues. Animal studies often lack allocation concealment, and also they lack blinding. And the lack of randomization of animals into different houses and different cages would often conflate and confound outcome assessment. And therefore, it is hard to assess whether these studies have an intrinsic high or low risk of bias. In conclusion, through the use of behavioral measures such as MMSE, wire maze, Morse model maze, and also passive avoidance, we have found that there has been a significant association for probiotic supplementation and improved neurocognitive function, despite the heterogeneity and bias found amongst the studies, and despite age being a confounder. The biomarker analysis in qualitative results supports the role of probiotics in ameliorating age-related neurodegeneration, because it shows that these pathways are indeed upregulated, and these pathways indeed exist to try to curb Alzheimer's pathology. However, the unclear risk of bias in both human and animal studies suggests that there is a need for more robust human randomized control trials to support its use in clinical practice, and currently its benefit in humans at least is rather dubious. And nevertheless, with probiotics showing an effect, there's always some kind of hint as to whether prebiotic supplementation towards probiotic use would be beneficial as symbiotics. I would like to thank CUHK and Cambridge for letting me do this research project. And this is the end of my presentation. I'm happy for any questions.
Thank you, Christopher, for your very wonderful talk. So I'm now taking questions from both Zoom and YouTube. There are a few questions on Zoom. First one, what are the differences between M1, M2, and further phenotypes?
M1 phenotype of macrophages is a pro-inflammatory phase. M1 phenotype for macrophages and microglia are pro-inflammatory. It is often associated with interleukin 1 beta, interleukin 6, interleukin 12, interleukin 18, etc. M2 phenotype of macrophages and microglia are associated with interleukin 10, TGF beta, and interleukin 1 receptor antagonist, IL-1RA. And essentially, it's an anti-inflammatory phenotype. And in the case of Alzheimer's disease, it's oftentimes that the M1 phenotype is upregulated too much, such that there's chronic inflammation. And the other phenotypes, which are not defined by the M1 and M2, regulate other minor pathways, but that doesn't concern us here. And in fact, the M1 and M2 polarity is often oversimplified, and they do not represent the whole picture of the pathogenesis in the brain.
Thank you. Second question, what sort of probiotics are being used? Are there other researchers trying to promote particular species?
Yes. The current dominant paradigm for probiotics include various strains of various strains of the Bifidobacterium genera and also the Lactobacillus genera. For example, look at here. There's use of Bifidobacterium longum, lactis, etc., and Lactobacillus like Plantarum, Saysi, Ramosus, Pentosus, etc. However, and both of these are proven to be quite effective, like the Yakult drink, and also even in yogurt and kefir. Lactobacillus is the most common probiotic strain. However, in recent years, there's some research into the use of Clostridium, bacteroides, and even Enterococcus as to see whether they have probiotic benefits. However, the research into these species is kind of limited. There's also some research into the use of selenium-enriched yeast. However, the research on that is also quite limited because to survive in the gut, you have to either be a facultative anaerobe or a complete obligatory anaerobe, and it is hard to survive in this species. And also to survive supplementation via the gut, essentially passing through the stomach and also the intestine metabolism, it has to be supplemented in a way that it won't be killed, destroyed by the stomach enzymes and stomach acid.
Thank you. One more question from Zoom. Would you mind elaborating on LPS-induced neuroinflammation? Sorry.
Yeah, sure. Let me find a slide. Okay, I found it. I can't find it. Doesn't matter. Essentially, LPS is referred to lipopolysaccharides, and they are a hallmark of gram-negative bacteria, and particularly in gut dysbiosis-mediated enhancement of Alzheimer's pathology. LPS induces localized inflammation in the gut mucosa, and this results in increased leakiness, increased permeability of the gut mucosa towards the gut lumen. And this increased permeability due to LPS allows LPS to enter the bloodstream, the circulation at a subclinical concentration, and ultimately through the bloodstream it travels towards the blood-brain barrier, cross it, and into the brain CSF. And the LPS in the brain CSF, despite being in very low concentrations, is able to induce low-grade chronic neuroinflammation, and therefore driving M1 microglial activity, and ultimately contributing to further exacerbation of neuroinflammation, and ultimately contribute one small part towards the entire big picture of Alzheimer's disease.
Thank you. So there's one more question. Have there been any papers testing a multi-pronged treatment with probiotics and other treatments together?
Oh, yes, certainly. In fact, most of the studies don't do it merely on probiotics, since it's unethical to just give Alzheimer's patients just probiotics, and also in animals. Probiotics per se is used. However, probiotics with exercise, probiotics with acetylcholine, probiotics with, I forgot what drug is it. It's like anti-Alzheimer disease modifying agents. I forgot the exact drug. And also probiotics with some kind of anti-inflammatory drugs is also used. And also in recent ages, there's also probiotics in prebiotics, prebiotics being fructooligosaccharides, which essentially are substrates for probiotic bacteria strains to further increase their metabolome profile. And all of these are currently being researched. If you do a Google Scholar search for them, you'll find that many groups in Japan, some in China, some in Europe, and also some in Iran are doing it right now. However, the quality of data in these studies are a little bit dodgy, particularly in the Japanese group, since the Morinaga Milk Company, which is a confectionery company, they've been funding studies for the use of probiotics and prebiotics into whether they improve neurodegenerative pathologies. But yes, there are certainly studies and a multi-pronged approach used, and there are plenty of animal and some human data on it.
Thank you very much, Christopher, for your very elaborate answers and informative talk. So if there are no more questions, I will summarize this session. So thank you again, Chris, for presenting different hypotheses of Alzheimer's disease and data on the effects of probiotics on Alzheimer's, and also assessing the quality of those research. So next up, we at 2 p.m. we'll have a medical physics session, which may be of interest to neuroscientists, because one of the talks will be on brain imaging of anesthesia. And at 4 p.m., we'll have our final keynote speech by Dr. Rolf Weller, who is the director of the European Bioinformatics Institute. And the last session today will be on neurobiology again, and it will be the last session of the entire Varsity Sci symposium. So hope to see you all again soon. Thank you.
Okay.